Bismillahirrahmanirrahim. Hormone secretion and transport are discussed in this video. The hormone secretion after a stimulus and its action duration is different for different hormone. Whenever there is a stimulus, the hormone is released and a specific action of that hormone is produced. In some hormones, these hormones are released within seconds, and their action lasts for seconds and minutes. And the example of these is non-epinephrine and epinephrine. While for some other hormones such as growth hormone and thyroxine, they are released within seconds to minutes after the stimulus, and they exert their action for months, and these specific functions are tailored to perform a specific control or a specific effect of that hormone on the body. Here an overview of the hormone secretion and action timeline is given. Catecholamine, that are epinephrine and norepinephrine, their action onsets within seconds and duration is 4 minutes, and they are for rapid fight or flight response. The peptide hormones such as insulin, glucagon, and antidiuretic hormone, their onset of action is within minutes and the duration is uh, minutes to hours, and they are responsible for metabolic regulation and water balance. Steroid hormones such as cortisol, aldosterone, and testosterone, their effect onset is for hours after stimulus, and their duration of action is hours to days, and they are responsible for sustained metabolic and electrolyte control. Thyroid hormone, the amine group of the hormone, triiodothyronine and thyroxine, their onset of action is hours to days, and their duration of action may last for months, and they are responsible for long-term regulation of metabolism and growth. Growth factors and long-acting hormones such as growth hormone and insulin-like growth factor, the onset of action for these is hours after the stimulus, and they exert their effect for days to months, and they are responsible for tissue growth and cellular regeneration. So there are fast acting hormone like epinephrine that operate in an acute stress and emergency scenario, and then there are slow acting hormones like thyroid hormone or growth hormone that ensure chronic stable control of physiology, that is the growth and metabolism. The nature of the hormone such as the chemical class uh, influence its solubility, transport, receptor type, and hence the action profile. The concentration of the hormones in the circulating blood is incredibly small. They are about 1 picogram, or that is about 1 million of 1 million of a gram per milliliter of the blood, or microgram, that is 1 of 1 million of a gram per milliliter of the blood. And these small quantities of the hormone are responsible for highly specialized mechanism in the target tissue. So these small quantities may exert a powerful control over the physiological systems. Here in this table, the hormonal concentration and secretion rates are summarized. The hormone concentration is about 1 picogram to a few microgram, and the secretion rate is about microgram to milligram per day. So the unit is microgram to milligram per day, and even At such low output, the effects are physiologically potent. For example, the thyroxine is about 8 microgram per day secreted, and its serum T4 unit, T4 level is about 0.08 microgram per uh, ml, and it supports the basal metabolic rate across all the tissue. Such minute quantity support whole body BMR. Then insulin, that is about 40 to 50 units per day secreted, that zero point 0.5 to zero point 0.6 microgram per kg per hour. Its unit is 5 to 15 micro unit per ml of serum. It modulates glucose uptake minute to minute within the body. Another example is estradiol, that is secreted about 50 to 400 picogram per ml during the menstrual cycle, and its unit is picogram per ml, and it regulates the reproductive and secondary sexual characteristic in female. So why such low quantities are effective? High affinity receptors in target cell, they ensure that even picogram levels can initiate a strong response, a strong biological response, and there is also a process of signal amplification through second messenger systems, for example, cyclic AMP and calcium, that magnifies the effect. Then there are presence of efficient feedback loops, that for example, the negative feedback in hypothalamic pituitary axis that tightly controls the secretion. So ELISA, or enzyme linked immunoassay or radioimmunoassay, are used to measure the levels of the hormone, and this procedure must be ultra sensitive due to these low concentration. The pathological states like Cushing syndrome, Addison's disease, or thyroid storm result from a small quantitative deviation that results in large physiological consequence. Thank you for watching this video. If you have any query, please write in the comment box.